Hello, my name is Sabine Fischer and I'm a postdoctoral researcher at the University of Frankfurt. And during my work I use mathematical methods to figure out cellular processes. In my talk I will show you how we used Mathematica to build an image and data analysis pipeline and how we applied it to analyze cell packing in three-dimensional cellular aggregates.
Hi there. We are going to start our next presentation from Sabine Fischer, and her presentation is called Mathematica and Cell Biology, Image Segmentation and Analysis of 3D Tumor Spheroids.
Thank you. Hello everyone. Thank you for coming to my presentation. I'm going to tell you a bit about how we use Mathematica to do research in cell biology, so to understand cell biological processes a bit more. I'm a postdoc at the Goethe University here in Frankfurt, and I'm part of the physical biology group. And the great thing that I love about this group is that we are a very heterogeneous group, so we have people from all sorts of disciplines. We have biologists, chemists, physicists, mathematicians, bioinformaticians, everything almost you can think of in natural sciences. And we're part of the biological science department. And what we do is we look at cell and developmental biology. We do quite a lot of microscopy, also microscopy development. And then, of course, we need to do data processing and try to do quantitative analysis. And essentially, what our idea is that we try with the whole group, we try to match this whole circle. So everyone, of course, needs a scientific challenge to start with. But then we have the methods development, for example, the microscopy development. We have the experiments, the traditional ones done by the biologists. And then, and that's why it's called physical biology group, we really want to do quantitative analysis of our data. And this is where I come in, and this is also what I'm going to tell you about today. So this part down here, the data acquisition, more or less the data processing, data analysis. And then, and some of our questions, we also do some mathematical modeling, but I'm not going to talk about this today. And then, of course, you need to reconsider, go back to your scientific question, and the whole cycle starts again. Okay, so what's the scientific question for today? What I'm going to tell you about is multicellular spheroids. So these are three-dimensional cell clusters, and they essentially form, you put the cells in a well, and then they aggregate and form 3D clusters. And this is what you'll see in the movie here. So on the left, you see the wide field image. On the right-hand side, you see a fluorescent image where we've tagged the cell nuclei. And essentially what we look at the well from the bottom, and we see how the cells come together. And then you see, because it's just a 2D image, if it gets more and more blurry, that's when the cells go in 3D, and you end up with a real proper 3D ball. And I'll show you what this looks like in the next slide. So here we'll see, I'm just going to pause that for a minute. So here at the bottom, we see this is our 3D spheroid imaged with a 3D microscope, and we've just cut out a bit to demonstrate what it looks like. And on the top, you see one slide out of this spheroid. And spheroids are very popular models for tissue studies and also tumor studies. So in this case, the spheroid is formed of tumor cells. And why are they so popular? It's because they resemble typical features that you see in tissues. So in this slide here, we see that we have three different zones. So in the middle here, or also you can see it on this image here, the cells die. There's a necrotic zone because this spheroid is rather big, so nutrients like oxygen can't get in. Then here we have a quiescent zone where the cells essentially just sit, they don't do much. And out here we have cell division going on. So we have quite a good representation of an avascular tumor, so a tumor where you don't have vasculature yet. And this is why they're very popular to sort of study, for example, drugs, because in former times what people used to do is they do a 2D cell culture, so you have like a glass dish and you put the cells in and then you test your drugs. But we are not not two-dimensional, we are three-dimensional. So, and people have found out that to actually study cell interactions in 3D, you get different results, and you also get different results from the drug testing if you do it on 3D spheroids instead of these 2D tissues. Okay, so so far, what most people do is they have the spheroid, and then they put a drug on it, and they see whether it shrinks or not. They just look at it in a 2D from the top, and then they look if the diameter shrinks or not. But with our fantastic microscope, if you look at the image here on the side, then you see these individual points, and these are the cell nuclei. And if I let it run again, then you can see that we can image the nuclei through the whole spheroid. And so we have the opportunity that we can actually measure what the individual cell does in these spheroids, which most people can't. So our goal, what we want to do is, we want to use these spheroids to analyze the drug effects, not just on the whole spheroid, but really on individual cells. And for that, what we need is an image and a data analysis pipeline, because we need to be able to automatically detect the cell nuclei and see what their distribution is within the spheroid. Because with these ones, they have, 35,000 cells, so you really don't want to do that manually. You don't want to click on every single nucleus. And this is what I'm going to tell you about today. I'm going to tell you about the data analysis pipeline that we've built up, and I'm going to show you one brief result for that. Okay. Now the problem is, of course, Mathematica has all these nicely built-in functions for image analysis, and one of them is that you just do a binarization. So you do a global thresholding. Every pixel that is above a certain value is foreground, and the rest is background. If we do that on our spheroids, then we get really densely packed clusters. So here we see on the image on the right, we see that different components that belong together, they have the same color. And we can already judge that here, for example, this area, that is more than one component. You can see it must be 3. But this just simple segmentation doesn't really work. So we'll have to come up with something better. So what we've developed is this 3-dimensional cell nuclear segmentation pipeline, let's say, approach, where we combine 2 different ways. So we start, this is our image stack that I've showed you earlier, where we just go slice by slice through the 3D volume. We can put it together into a 3D volume, and we use image 3D in Mathematica for that. And then we do 2 things. One thing is we do this binarization, as I told you in the slide before, so you detect foreground and background pixels. But what we also do is, to split up the ones that are clustered together, we find seeds within the different components. So our seeds should represent different points, and each seed should represent one nucleus. And then we can use the seeds together with the binary image, and then we get a components image where we have them all separated. And out of that, we can extract the features. So I'm going to tell you a bit more in detail, and I'm starting with the binarization. So what we do, we don't quite do the global binarization that I've just talked about, where we use the local adaptive binarization. They've implemented that recently, and it's quite useful. You don't do a global one anymore. It essentially separates the image into individual parts, and then it does the binarization on every single part. So if you have variations in local intensity, those are matched. It's adjusted to that. And this is essentially now one problem with the local adaptive binarize is that if you do it in 3D, it still runs forever. So what we've decided to do is we do it in 2D along the different axes, and then we put the picture together. So we sort of map it over all the slides, and this is what this 3-way local adaptive binarize here essentially does. So here we have it 3 times, 1, 2, 3, and then we put the image back together, and that works quite nicely. So here I can show you an example on how it works on a stack. And so here we see the 2D slices, and on the right-hand side here we see the outlines of the cell nuclei that it has detected. So if we look along this axis, it all looks very good. We can already see that it's recognized the images very well. However, if we put it all together and show it as a 3D image, we see this. And remember, one color is one object, so here all the cell nuclei, they are all still clustered together. So what it's done nicely, it has nicely separated the spheroid from the background, but it wasn't very good at separating the objects. And if we look at that in detail, I'm just going to give you 3 examples. So here are clusters that are just clustered together. So here we have 2 nuclei, 3 nuclei, 4, this is a whole cluster. And that is just one object at the moment. So far this doesn't quite do what we want. We still need to separate our clusters. And what we do is we use a seeded 3D watershed. So as I said, we use seeds to identify where a cell nucleus should be. And then, for example, if we look at this cluster here, what we will get is we will get 3 positions. And out from these 3 positions, you have sort of like water spreading. This is what the watershed algorithm does. You have the water spreading, and it will spread from this region outwards, from this region outwards, and then where they meet, sort of these water basins, that's where you get the separation between the objects. But first of all, we need the seeds. So for the seeds, what we use is we use the Laplacian of Gaussian filter. It's again a function that is implemented into Mathematica, but we don't use it just in this simple way because that doesn't quite do the trick. What we do is we use it at different scales. So if you think of a Gaussian filter, it usually filters the image by taking sort of looking at the image, the pixel values in a certain region or the voxel values in a certain region around the voxel, and then computing the new pixel from that. And this is what is meant by the scale. So for the Laplacian of Gaussian filter here, this is again our function to detect the blobs. What we do is the main part is here. We use a map, and we do that over a certain range of scales. And then in here, we use our Laplacian of Gaussian filter, and this is where we use the scales. So if you have a smaller scale, you will detect smaller features in your image, and if you have a larger scale, then you will sort of detect larger blobs. It's a bit like zooming in and out on an image using these different scales. And then these other parts, these parts here, they sort of just they take the voxels and look at which scale you get the highest response from your Laplacian filter, and that's probably the part where you have the blob. And if you do this, we do that on our example image that I showed you earlier, then this is where we get the seeds. Now I've dilated them a bit. They are usually smaller, but so that you can see them on the screen, I've enhanced them a bit. And we can see that we get nice positioning of the seeds on the individual cell nuclei. And if we look at it in 3D, then here we have this nice sort of cloud of points, essentially. So this gives us just the position of our nuclei. It doesn't give us the volume or the extension yet. So to get the extension, we then combine the seeds with a 3D watershed. So as I explained already, it sort of, you drop water where the seed is, and then it expands, and that will give you the regions of the nuclei. This is the result for our example image. And now we see that we have the nuclei nicely separated. So we have the whole areas, or in this case, it's 3D, the volumes. And if I go through the slides, we can see that each color represents a nucleus, and it matches the original one quite well. And compared to earlier, if we then look at it in 3D, we also see that we have the individual nuclei now, not just the whole blob in one color. And just to visualize it a bit more, I'm going to show you my examples from earlier. So these were the ones, they were all combined together, and now we have 2 nuclei here. This one's also nicely separated. Here we have our big cluster, again, that worked rather well. And there we have the 4th example. So there you can also see you spread out from here, and then this is where the 2 water basins sort of meet, and this is where you get the separation. Okay, so now we know where our cell nuclei are. We know which voxel belongs to which nucleus. So we've basically finished the segmentation, and we can now move on to extract the features. And of course, first of all, there's the built-in function from Mathematica, the component measurements. And there we already get quite a long list of things out that we want, and we save it in this sort of option format because then it's easier to process it later on. That's the funny thing. Okay, so for example, we get the label, we get the centroid, the count here is the volume, intensity centroid, different measures of the intensity. But there are other things that we would like to know for our purposes. One thing, for example, that we want to know is does the property of a cell nucleus, does it depend on where it is in the spheroid? So what we do is, of course, for the spheroids, we only have the cell nuclei, so we don't really have like the whole boundary of the spheroid. So what we use is we use the spherical harmonics. So we've heard them earlier today, some of us maybe. Here we use them in a completely different context. So the idea is that to use these spherical harmonics sort of as building blocks, like Lego pieces maybe, to approximate the surface of our 3D spheroid. And some people have actually done it a bit extensively. They've tried to rebuild to approximate the head of Max Planck with the spherical harmonics. And what we see here is you have them in different orders. So that's essentially the building blocks you use. If you have a higher order, use more building blocks. And you also get more detail. So here they've used an order of 2, and then 20 and 50, where they get really precise detail. Of course, for our purposes, the 20 and 50, that will only give us artifacts, because our spheroids are rather round, so we might want something that's close to an ellipse, maybe. So to use these, we use our positions of our cell nuclei. So this is what we do here, centroid. And then we can just use the replace. That's the nice thing about having those rules that I said earlier. Okay, and then we can plot them, and we have this whole cloud of thing. But to actually use the spherical harmonic approximation, we only need the surface points. So we don't want the whole cloud, we want the surface of it. And this is where I'm going to show one example of how to get a mesh, and I'm going to show you a different example later on. And this is sort of the older version. So one thing that Mathematica has built in is this TETGen link. So it's essentially a link to an external program that they found might be useful, and it's called this tetrahedral mesh generator, and it was developed at this institute in Berlin. And with this TETGen, we can generate a convex hull. Of course, with the new region functions for Mathematica, you can probably do that now with the Mathematica built-in functions, but when I did this, they didn't have them yet. And from this, we can extract the surface points, and then we can plot the surface points, and we see that here, this is just the surface. And now we can use the spherical harmonics functions as before, as my colleague said this morning, the problem is they're not real functions, so I'll have to adapt them a bit so that I get a real surface. And I can do all these calculations, and then I can plot them. And here we've chosen an order of 3, which is sort of a sensible value for our purposes. And this is now our point cloud with the surface built on top of it, and we see it fits it very nicely. So now we have ideas about the whole spheroid, and we can measure things in this whole spheroid. So we can measure the centroid, for example. Other things, so what I usually do is I don't choose one order, but I've chosen from order 1 to order 7, and then I look at which one's most appropriate. So here I have the approximation vector and also the approximation error for orders from 1 to 7, and I can also then calculate the volume, of course, of my spheroid, which I couldn't before. And once I have this approximation of the surface, I can then say more things about my nucleus, because then for each nucleus I can say where is it in terms of distance to the surface of the spheroid. And this is what we essentially add to our list. So again, this is our example nucleus. Of course, we will have that like 35,000 entries for that, but this is just one example. And to all the measures that we had before, here's the intensity. We now add the surface point. So here I've chosen order 3 is the best for that example case, order 3 or 4. And there we have a surface point, and we also have a distance to the surface. So now we can have a better idea of where each nucleus is in our spheroid. And then what we're really interested in is how well are these how are these cells packed? So how many neighbors do they have? What is the distance to their neighbors? And one good approximation of which cells are next to each other is if you use the Delaunay triangulation. So the Delaunay mesh gives you a triangulation where you have the interior angle is maximized. And it's usually, so many people use it to describe neighborship relations between cells. It's a very popular method for that and has been shown regularly that works quite well. And this is now where I'm showing you the second version of how you can get a mesh, because now I'm actually using the Mathematica built-in functions mesh, Delaunay mesh. And then we can extract so here we can generate the mesh from our coordinates. This is again our coordinates with the centroids. And then we can extract the lines from it here with this command mesh cells. And this is what would give you the image on the left. And there we see that particularly, maybe here we have still quite long distances, or we have some of these lines, they are actually spanning over several, this one here, it spans over this cell. So this is not what we would call a neighbor if you look at it with your eye. So what we then did is we actually deleted all of those lines that were longer than 30 pixels in our case, which is sort of a reasonable distance within our sizes of cells. And then we get the picture on the right, and that looks much more reasonable. So here the red parts are our cell nuclei. So it's just the positions, and I've put a sphere there. And then the white ones are the connecting lines. And with this knowledge, we can then update again our list of measurements for our nuclei. And this is what I've done here. So we append the neighbors, and this is where we determine the neighbors. These are all the functions that do it. But essentially here at the bottom is where we get the results. And I'm just going to add a few lines so that it's a bit better visible. Here we had our distances, and now we can count the neighbors. So we can count how many white lines go out of one of those red dots. And we can look at the distances to the neighbors, and we can also look at the standard deviation. Once we have all these measurements, what we do is we save them in a notebook. So we create a report, and I'm just going to quickly do this here. So here we use create document, and then we have a text cell, and then we essentially plot all our functions into a notebook, all our results, and then we can save the notebook. And this is what it will then look like. So we have a title and we have the date, so it's just been generated. And here I've just plotted 3 parts just for one example, but essentially we then have all the plots for all the different data sets, and we can go through them and we can check. And this is without formatting. This is just the raw data, so that we can look at it and check which one has interesting features, which ones do we want to look at further. Is there anything that doesn't look like we would imagine it to? Then we'll have to go back to the images and see if something went wrong. So it's a result, but also to check whether everything was okay. And then out of these results, we can then choose the most interesting ones and plot them. And I'm giving you one example here. So the 2 functions we're using here is the density histogram and the arrow bar plots. And what we want to look at is we have these 2 data sets. And in one we found 25,000 cells, and in the other one we found 35,000 cells. And you can see by eye already that the structures look somehow different. So we have the impression that depending on the cell number, you get differences of cell packing. So here you have a much larger necrotic zone. And this is what we've plotted here with the density histogram. So we've plotted we have the distance to the surface, and we've normalized it. So at 0 is right next to the surface, at 1 is in the center. And then we have the number of neighbors, and the color gives us how many % of cells have that number of neighbors. And we can see here is quite a high number of cells with about 15 neighbors, which is what we would expect from sort of a hexagonal packing. But in this area, we can also see that here is sort of an extra peak that we don't have here, where we have more cells that have less neighbors. And this is what we would imagine from this image as well. And if we use error bar plots instead, then this plot the function as it is here, then again we have the distance from the surface. So here's the surface, here's the center, and I've plotted the mean number of neighbors and the standard deviation, and there we can again see that here we have sort of a wider region where the cells don't have many neighbors. So essentially we now have a pipeline where we can put in all these data sets. So our colleagues, the biologists, they will then produce, I don't know, 10, 20 spheroids. We plug it, we put them into the pipeline, and then we get these graphs out. And we can then, of course, do that same thing with spheroids that were treated with drugs. So then we can find differences in the cell packing and see what effects certain drugs have. So to summarize, we've developed this Mathematica pipeline so that we can analyze the packing of the nuclei in the 3 dimensions. For the segmentation, I've used the local adaptive binarized, the Laplacian Gaussian, and the watershed. Then for the feature extraction, we've seen we have this list of replacement rules that is very handy. Use component measurements, the spherical harmonics, and these different types of meshes. And for the results, we first of all save them in a notebook, and we also have the density histogram and the error bar plots. And as I said, this is sort of the basis then to do drug testing on spheroids. So finally, I'd like to thank our group, the physical biology group, and in particular, of course, our boss, Professor Stelzer, and then Alex and Bina, who are sort of with me in the theoretical team and are also here in the audience. And finally, there's one little thing that I would like to mention is that we do Mathematica courses. So we run 1 week introductory courses, and they're usually in March. They're a whole week where we do mixtures of talks, and people can do their exercises, and some of the people in the audience have already been there. And we also do 1-day advanced courses. So, of course, image analysis and statistical data analysis are sort of our main topics at the moment, but we're also thinking of expanding them. And they're open to everyone who would like to come along. So if you're interested, you can either go to our homepage or just send me an email, and then I can keep you updated on the next dates. And that's it. Thank you very much.